Welcome to the WERA 103 Nutrient Management Lectures and Seminars. We're glad to have Dr. Joan Davenport as our presenter today. So we'll be speaking about how organic phosphorus fertilizers work and if they can be used effectively in field crop production. Before she begins, let me just give you a brief introduction to Dr. Davenport. Dr. Davenport is a professor of soil science at Washington State University, and since completing her PhD in 1985, Joan has worked at Washington State University and also at Ocean Spray Cranberries. And during her career, she's conducted extensive research on plant nutrition related to perennial fruit crops. In addition to her work at Washington State University, Joan has worked extensively internationally as well, particularly in Australia and Chile, working with wine grapes and cranberries. She's led an active research program and a teaching program. In her research, she's been influential in developing standards for tissue testing for cranberries and grapes, and continues to do that for the Washington blueberry industry. Joan, as I mentioned, is a soil scientist, and she has a passion for soils and soil management. And in this role at Washington State University, Joan has taught extensively, including subjects such as soil physics, pedology, soil fertility management, viticulture, and current topics in organic and sustainable agriculture. And she also developed a capstone course for the students at Washington State University in their viticulture and enology program. Joan has had an active research and education program. As I mentioned, she's received many awards and accolades for her both research and her education programs. She received recognition from the Washington State Grape Society with their Clora Award, and also she has been inducted as a fellow of the American Society of Horticulture Science. She's been active in leadership in many national and international organizations where she has shared her talents and her insights on managing nutrients for sustainable production of perennial fruit crops. So Joan is certainly well qualified to talk about this. She's been an active member of the WERA 103 committee, and we're glad that she graciously consented to present this overview of some of the work that she's been doing recently. So Joan, I'd like to turn the screen over to you, and then we'll get started with the presentation.
Thank you very much, Rob. And can you let me know when you see my screen? There we go. Oops.
Okay, if you can maximize that, we'll be ready to start. Great.
Okay, well, thank you for that very nice introduction. So I'm going to talk today about phosphorus fertilizers in general, and then talk about a couple of studies that we've completed recently, completed in 2017, looking at phosphorus fertilizers. In the introduction, Rob mentioned that I've done a lot of work on perennial fruit crops, which has been a large focus of mine. This particular project actually worked in field crops, and I do want to mention that the results of, or this project was specifically from my recent graduate student, Prudent Rampeja's PhD project. And so a lot of the credit for this work goes to Prudence. Her PhD dissertation is now published, and we're in the process of getting papers from this material published. So just as a quick overview of what I will cover today, I'd first like to go over a little bit of background information focusing on phosphorus and phosphorus fertilizers. Then I'm going to talk about the results of two studies that we did for Prudence's project. And I'm talking about them a little disjointedly in that I will first present the results from the field trial, which was actually done second, and then the results from the pot study just to cover some bases on soils, and that was done second, and then provide you a brief summary. Why phosphorus? Well, phosphorus is an essential plant nutrient. If we think about the macronutrients that plants require, the first three that are always spoken about are nitrogen, phosphorus, and potassium. And as essential plant nutrients, there are no substitutes for phosphorus. Plants cannot go through their entire life cycle and reproduce without adequate phosphorus. Phosphorus is important in many roles in the plant. It certainly is important in energy transformations. And so obviously any growing and developing plant needs phosphorus for that. In addition, phosphorus also plays a role as part of the backbone of all plant genetic material. So again, very, very important in plant productivity and growth and development. Without phosphorus, we can't produce food. And since agriculture exists for many reasons, including the production of food, this makes this a very important nutrient. I also wanted to mention that when we talk about particularly organic nutrient sources, which we're going to be speaking about a lot today, most of the work that has been done on these has focused on the other plant macronutrient, nitrogen. So this project is a little different in that we are looking at plants with adequate nitrogen supplied to them and focusing only on phosphorus. So what are some of the reasons that we see phosphorus as a challenge? Well, phosphate rock is a mined mineral, and there isn't enough, well, there's a limited resource for this particular mineral. And so we're concerned that the global resources for this mineral are becoming limited with our use of it in many ways for many things, including fertilizer. The other problem with this mineral is that it forms slowly over time. And at the rate we are currently using it, we won't have another 10 or 15 million years to necessarily create more of this mineral. I mentioned food, and food production is expected to need to increase by 70% by 2050, largely to deal with population growth. And if you look actually even from 1950 to the year 2000, we see this vast increase in people. And that's continuing. And all those people need food. But when we look at how much phosphorus production from the rock mineral we have, this is a curve. Again, look at that period. Here is 1940 and there's 1960. So looking at that period from 1950, and we saw that vast population growth, and concurrent with that, we see an increased demand in how much phosphorus production for fertilizer we've used, where the red line is the model and the black dots are the actual, but you see that it's going up. So then the question becomes, how can we provide food for this increasing population when this key nutrient that we need is actually decreasing in its availability or in the amount we have stored in the earth? So this would just be an overview of how we get this phosphorus mineral from a mine, and then we create it or turn it into a phosphorus fertilizer through a fairly intensive process. Then we apply it to the land, and then we harvest for feed for people and for animals. And then we have some leftovers. We feel very strongly that we need to consider alternatives to mineral phosphorus as phosphorus resources for food production, where we could take some of the waste byproducts from animal manures, essentially, and or vegetable-based products, and use these to recover the phosphorus and then land apply it. In the US, we have over 369 million metric tons a year of manure basically provided in feeding operations. and there's 13 more times sewage produced by the US population, giving us the number that you see on the bottom of this page of 365 million kilograms of phosphorus per year produced that we currently are only using a tiny fraction of. So again, how can we recover this? In the project that I'm presenting today, we actually looked at 2 different ways of recovering that phosphorus and making it available. We didn't actually do the recovering, but we looked at products that came from 2 different ways. One is called composting, which many of you likely have heard of. The other is called anaerobic digestion, which is a slightly different process, and let me go over both of those. So composting is, again, a process many of you are familiar with, where animal manures or vegetable materials or other items that are rich in organic matter and minerals are basically allowed to decompose by microbial organisms, and then there's enough water provided. But please notice that this is also a process where oxygen is involved. So it's an aerobic process where the output is the composted manures, which again have organic matter, minerals, and water in them, but typically by the time we're using them, they are stable. So this picture in the center shows you a nice pile of compost material, which looks like very rich soil and can be added to the soil as a potential fertilizer source. The anaerobic digestion process is a little bit different. And again, this is done with animal manures, where their carbohydrates, fats, and proteins are digested by bacteria. And then what's nice about this process is it can be used for 2 different purposes. You'll notice this bacterial anaerobic digestion can be used to create some biogas, hence heat and electricity. But then there's this leftover effluent. And what we do with this is we actually increase the concentration of the phosphorus and actually other plant nutrients like nitrogen in the process to make a highly available phosphorus and nitrogen fertilizer product. So in this study, what we did was we're looking at recycled phosphorus products to see if they could be substituted for our conventional fertilizers. So we felt that the things we needed to look for were crop response. Is the crop taking up material? Is it producing enough plant material? Are the plants growing well, which would be the biomass production? And also, because we're interested in food crops, and in this case, field crops, we are interested in yield. Another thing that we wanted to monitor is if the phosphorus release from these recycled or organic nutrient sources was consistent with the rate and availability that we would get from a mineral phosphorus fertilizer. And the reason for this is oftentimes when we're using organic nutrient sources for nitrogen, we only expect about 50% of the material or the nitrogen to be available in a single growing season. But we haven't really done the research on phosphorus to see if the availability is similar. So that was of concern. And we also did want to see some differences in different soils to see how that might affect things. So the 2 field trials that I'm going to talk about were done on 2 very important crops worldwide. One is corn, which I have a picture of on the left-hand side of the screen, and the other are pinto or dried beans, which I have a picture of on the right-hand side of the screen. And these are important crops for both human and animal feed. So let me talk first about our corn trial, where we were looking in the field for the corn response to our different nutrients. And please note that both of these trials were conducted at a farm in Patterson, Washington, and the soil that you're looking at is called a Quincy sand. It is a very sandy soil, so we don't expect a lot of nutrient availability. It's also low in organic matter, so we did not expect a lot of nutrient coming naturally from the soil. These were the treatments we had in our corn. So we used 3 different fertilizers, the anaerobically digested dairy manure, and I spoke about that process a little while ago, and that was a fairly high phosphorus concentration of about 30%. Composted chicken manure, which had a fairly low phosphorus content of only 2%, and then monoammonium phosphate, which has about 50% phosphorus in it. And we also looked at adding absolutely no phosphorus. This was done in a randomized complete block design over 2 growing seasons. And you'll notice on the right-hand side of the screen, we have the trial in 2016 and the trial in 2017, where we have a slight difference in our phosphorus rates. We always use a 0 control. And then the number that you see highlighted, the 20, was actually the rate of phosphorus in kilograms per hectare, which is pretty similar to pounds per acre. The rate of phosphorus recommended by the soil test for phosphorus for our area. So our other rates were half of that, or 1.5 times that, or 2 times that amount. Based on our results in 2016 and 2017, we wanted to see if we went with a very high rate of phosphorus, how that would impact things. So we actually did not use the half rate in 2017. We did a lot of sampling to determine the phosphorus uptake or utilization. So we actually monitored tissue nutrient throughout the growing season, and we used the 3 corn stages of V6, V11, and VT, where V6 and V11 are 6 and 11 leaf stages, and then VT is when tasseling begins. So that's just a shift in production. We also collected soil samples every time we took plant tissue samples, and then at the end of the season to see if there was any residual phosphorus in the plots. Again, we also monitored yield. Here's a nice picture on the lower left-hand corner of the corn as we were harvesting it. So we went into the middle rows and harvested a known amount of corn and weighed it so we would find out what the yields were, and we weighed it both with and without cob to figure out the actual yield of grain. We also then took a subsample of that and monitored that for how much phosphorus went into the grain itself. So let me talk first about uptake. This is our corn leaf phosphorus over time. And one of the things you'll notice is, regardless of the year, 2016 shown in the black dots and 2017 in the open circles, we noticed that there was an increase in the phosphorus in the corn leaves, showing an increase in uptake with time, which makes sense because the plants are getting bigger. But you'll also notice that in both years, by the time we get to the VT stage, our leaf tissue P is decreasing. Now that probably does not indicate a decrease in actual P uptake, but indicates a shift in where the plant is sending its phosphorus, because by the time we're going into tasseling, we're changing to the reproductive stage of growth. And so now the phosphorus is going to the seed, which we monitored later in the season. How did things respond to our different fertilizer treatments? Well, interestingly enough, in 2016, the only time we saw a significant change from the control, or the 0 phosphorus rate, with uptake to any of the products was with our composted chicken manure in 2016. Now remember, that's the year we had slightly lower rates and didn't have the highest rate. But with the anaerobically digested dairy manure and with the mineral monoammonium phosphate, even at the highest, even at twice the recommended rate, we didn't see an increase in our corn leaf phosphorus concentration. We were successful, however, in 2017 by increasing that rate, and so we've definitely felt like we went over the top. The other thing that I think is interesting in 2017 compared to 2016 is, again, with our composted chicken manure, when we go up to that 40 pound per acre rate, we definitely see an increase, although interestingly in 2017, that increase was only higher than the 0 phosphorus rate. In 2017, actually, with our monoammonium phosphate, our 40 pound per acre rate was also higher than the 0P, although not necessarily than the 20 pound per acre rate, which is the recommended. Soil test phosphorus was interesting, and again, we had some differences by year. The amount of phosphorus that was in the soil on average, actually, this is the amount of phosphorus in the soil at the end of the growing season. And you'll see with the higher rate in both 2016 and 2017, we had more phosphorus remaining in the soil at the end of the season than we did with the zero control. You'll also notice that in 2016, we still had a little bit of phosphorus remaining in the soil, versus in 2017, we utilized most of it. This probably was a difference due to the difference in temperatures and weather in the growing season. And we also did have an interesting thing in 2017, where at our 1.5 times rate of monoammonium phosphate, we also had high residual soil phosphorus. So what about yield? And yield is very important to us. This is a statistical table of our results on the top, and I'm not going to go through it in detail, but it shows you the different things we measured, which are the number of cobs, the weight of the cob, the dry weight of the grain, and then the phosphorus in the grain. And you'll notice that the only factor that made a difference in these was not the rate of fertilizer, was not the type of fertilizer, but it was the year. And you'll also notice that the yield in 2016 was higher than the yield in 2017. Not a lot, but it was different. And then the other thing that was also interesting was the phosphorus in the grain in 2016 was lower than it was in 2017. Now, if you'll remember our post-harvest soil test, you'll remember that we had less phosphorus in the soil at the end of the season in 2017. So this really shows us that we actually probably had more efficient uptake that year. And again, likely due to weather conditions. So just a quick summary from this. We definitely saw that the leaf P was higher when we increased our application rate, and our concentrations didn't really matter what material we used. So that really tells us that all of those fertilizers were equally effective. And also those leaf P concentrations tell us that the uptake by the plant was consistent. So even though we were using organic sources, they were quite available to the plants. Also, there were no yield and grain phosphorus responses to our different materials, again telling us that the organic nutrient sources were equivalent to the non-organic sources. Okay, so let's go over what happened in our pinto or dried beans. And you'll notice that this set of treatments looks very similar to what you saw before. Again, we took a pre-season soil test, so we used the same fertilizer materials, including a no P control. And remember that the composted chicken manure had the lowest overall phosphorus concentration, the dairy manure second highest, and the monoammonium phosphorus the highest. because beans have a lower nutrient requirement, their total amount required per growing season was, based on soil test, 12 kilograms per hectare. And so again, we used that half rate, 1.5 rate, and twice rate in 2016. But in 2017, we actually went up to a much higher, or 4 times rate. Similar design. One of the differences was that we actually looked at 2 different cultivars of pinto beans to see if there was a difference in response based on those cultivars. The plots were the same size. The tissue sampling, again, at 2 vegetative stages and 1 reproductive stage. And again, we took soil samples every time we took tissue samples, as well as post-harvest. Again, the harvesting was done from the 2 center rows. We calculated yield, and again, we also took a subsample of the seed and analyzed that for phosphorus to see how much went into the seeds. This shows the uptake this shows some things going on with the uptake, which really differed between the 2 growing seasons, which was surprising to us. So again, notice in 2016, and the 2 different colors are 2 different varieties, but notice in 2016, we see a slight difference in our uptake rate, or the amount, whereas it seems to really reduce in the leaf concentration by the reproductive stage in Buster, but not so much in Stampede. And again, we noticed that the seed pea concentration didn't differ, but just slightly in 2016. 2017, we actually had a slight increase in leaf phosphorus concentration. So all in all, this is telling us, regardless of the year, we are getting good phosphorus uptake. Again, our leaf pea data based on our different fertilizer materials and their rates, and this is 2017 only. The results in 2016 were consistent, except we didn't have that very high rate at the end. But you'll notice that it wasn't until we got to the highest concentration of fertilizer that we actually saw a difference from the control, at least statistically, although numerically we typically saw a slight increase in uptake regardless, as long as we were putting phosphorus fertilizer on. Soil phosphorus was interesting, and again, similar to with the corn, what we saw was that the soil phosphorus was continuing to increase even up until the R1 stage in 2016, but it did decrease, but not to as high as the post-harvest or as the pre-harvest level in 2016. But in 2017, we saw a pattern which would be more consistent with what we expect for uptake, where by the time we got to post-harvest, again, there was a lot less phosphorus in the soil, again, indicating to us that we probably had better overall uptake in 2017. Again, notice that F for fertilizer rate, fertilizer material, and R for rate did not have a significant influence in both years on our soil test P. Similar to what I showed you before for corn, for seed yield, again, we see that the only thing that made a difference was the year, or the cultivar we were looking at. And what was interesting was that in 2017, both cultivars yielded about the same. And so Stampede, which was down in 2016, was actually equivalent in yield to Buster in 2017. But fertilizer rate didn't affect anything. Fertilizer material did not affect yield. So the summary from this was that there was similar uptake based on what we saw in the leaves from the composted chicken manure and the MAP. The higher uptake of phosphorus was found in at the higher application rates, but we didn't see that in yield or in the phosphorus concentration in the seed. And again, we did see that there was slightly more translocation of phosphorus in Stampede than in Buster. So there can be slight differences by cultivar. So putting the two studies together, what we see is, again, regardless of the crop, our composted chicken manure and MAP gave us similar uptake of phosphorus. And in the case of corn, the dairy manure also worked as effectively. There was higher uptake when there was higher higher rates, but it didn't influence our yield or our phosphorus in the seed, telling us that the alternative fertilizer materials that we were using were as effective as that mineral phosphorus source, which is rather important to us, because again, if we're looking for alternatives, then this study really clearly shows that we can have as effective a treatment or effective response to phosphorus when we provide it through our renewable and constantly generating manure-based materials. So that was all a study on one type of soil. And we did start out the project thinking, we wondered about different types of soils and how they might affect the availability of nutrients. So in this case, we actually tested the soil of did a soil test on 5 different soils, and then applied just one rate of phosphorus or no phosphorus to all of those to see how corn would grow in a pot study. So the 5 soils we used varied by texture and also by soil pH. We had one soil from Texas, and that was is a clay soil called a Houston black soil. And then we had 4 soils from Washington, one of which was the Quincy sand, which I spoke about in the previous study. And then we had a Worden silt loam, which is a very commonly used silt loam in central Washington. In eastern Washington, we used a Palouse silt loam, and from western Washington, we used a Skagit silt loam. And what was nice about this was the characteristics that we got, which were different, were that the Houston black clay soil and the Worden silt loam both had slightly high soil pHs, plus differences in texture. And then the Quincy loam, or the Quincy sand, is a sandy textured at near neutral pH. And then the Palouse silt loam was a slightly lower pH, and then the Skagit silt loam even lower pH. So we were looking at how texture and, again, how pH could influence that phosphorus availability. We used the same fertilizers with the exception of adding a vegetable composted material to this, which was, again, fairly low in phosphorus concentration. And you see a picture of that in the left-hand, on the lower left-hand side. And then the picture on the right is that anaerobically digested dairy manure. We actually measured shoot biomass and root biomass throughout at many different growth stages. We also looked at soil test phosphorus, and we just were looking to see how the plants grew and developed, and if there was an impact of those different fertilizers, and if one was better than the other. I'm going to present far fewer results on this, but what was interesting was that plant biomass, the growth and development of the plants, showed some difference early in the growing season in response to the phosphorus fertilizers. And essentially when we had no phosphorus or the vegetable compost, we had lower phosphorus, and we also had lower vegetation. But by the end of the growing seasons, there were no differences in relation to the phosphorus fertilizer. Now, I need to point out that we actually had very few corn plants that actually produced corn grain. So that probably is the reason that we didn't see as much difference in the relation to phosphorus fertilizers. What was important, though, and I think this maintains that point that I'm trying to make, was the type of soil had more influence on the factors measured. So we had differences by the type of soil rather than by fertilizer, lack, either no fertilizer or any of our types of fertilizer. Again, telling us what we saw with the field study, that our organic sources and our mineral sources can be equally effective. So putting the two projects together, our summary is the vegetable-based materials did not work as well as we were hoping, but they were fairly low in phosphorus. However, those animal-based alternative P sources were as effective as mineral in field crop production, and that's very important because it tells us that these can be utilized. I spoke at the beginning about one of the differences we found when we're using organic nutrient sources for nitrogen versus our mineral sources, and that is that typically we only expect about 50% of our nitrogen to be available in a growing season when we're using an organic source, whereas we expect more like 100% or 75 or something when we're using a mineral source. However, we did not find that with the phosphorus. And so the nice thing is that our rate study really clearly showed that we don't need to use a higher rate of phosphorus when we're using our alternative sources. We can use what our current soil test recommends. There were a lot of people involved in this project. As I mentioned at the beginning, Prudence Rampesia used this for her PhD project. Many people from our sampling lab or from our soils lab came and helped with the project, and they all need recognition. So they're listed on the left-hand side of the slide. Other important things, the USDA ARS was very kind at helping us get things set up and using their field site. The research was also conducted at our Irrigated Ag Research and Extension Center. Very importantly, we had a collaborative relationship with Soil Test Farm Consultants who helped with a great deal of the project. And Prudence was also funded by a Fulbright Fellowship and the South African National Research Foundation, as well as Washington State University, to complete her doctoral program. So it's very important. It was a very important and interesting international and national collaboration to complete the project. Thank you for your attention to the presentation, and I would be happy to answer any questions if there are any.
Thank you, Dr. Davenport. We appreciate your presentation today, and people that are interested in contacting you for additional follow-up will be free to do that, and we'll be looking forward to having you publish the results of these interesting studies in the months to come. So thank you, and we encourage everyone to look at the other interesting topics on the WERA 103 YouTube channel, and I'm sure you'll learn a lot about soil and water management. Okay, thank you, Dr. Davenport, and goodbye.
Thank you, and have a wonderful day.